Okay, let's deep dive into some really fascinating research on treating canine brain tumors. We're looking at a clinical trial using a modified virus.
Oh, this is exciting stuff.
It is. They used a genetically modified oncolytic Newcastle disease virus, that's NDV for short. This specific virus is called rLAS-uPA.
I see. And what makes this research so interesting is that they use dogs who, well, develop these brain tumors naturally, and these tumors, they often resemble those seen in humans. It gives us a glimpse into potential human therapies.
And it's not just any brain tumors either. This trial, it included meningiomas, gliomas,
Oh, wow.
and choroid plexus tumors. All of these, they're prevalent in both dogs and people. So the potential implications are, well, pretty broad.
Absolutely. This study, they wanted to evaluate how safe this modified virus is, how the dog's immune systems reacted to it, and, most importantly, how effective it is in, well, either shrinking or at least stabilizing these tumors.
So safety first, right.
Yeah.
Tell me about the safety profile of rLAS-uPA. What did they find out?
Well, they used a very careful process called dose escalation. This helps determine the maximum tolerated dose, or MTD, and they found that most dogs only had mild to temporary side effects.
Like what?
Like fever or a little bit of diarrhea.
Okay, that's reassuring.
Yeah.
So what was the maximum dose they could give without causing, well, major issues?
It turned out to be 2/107 PEF/ml. And it's important to mention that only 2 dogs who received a much higher dose had more serious adverse events.
Okay, so safety-wise, it looks promising. But how exactly does rLAS-uPA work against the tumor cells?
It's really quite clever. rLAS-uPA is engineered to target a very specific receptor. It's called UPAR. And this receptor, you find it in higher concentrations in many types of canine brain tumors.
So basically like zeroes in on the tumor cells.
Exactly. And when the virus binds to this UPA receptor, it triggers a chain reaction. This causes the tumor cells to fuse, forming these giant cells called syncytia. And this, well, it leads to their destruction.
Wow, so it actually makes them fuse together. That's incredible. Did they confirm that the virus actually got to its target?
They did. They found viral genetic material in tumor samples that they took after the treatment. So yes, this confirms that the virus was infecting those tumor cells.
Okay, so that's a good sign. What about the results, though? Did they observe any tumor shrinkage?
Well, it's a bit of a mixed bag. Only 2 dogs actually showed significant tumor shrinkage. However, a good number of the dogs experienced what we call stable disease. That means their tumors didn't grow significantly during the study period.
That's still a good thing, right? I mean, stable disease for an extended period, that's got to be meaningful for dogs with brain tumors.
Absolutely. It can be a very significant outcome clinically. In fact, some dogs who didn't technically qualify for tumor shrinkage, well, they still showed a pretty substantial reduction in tumor volume.
Interesting. Now, what about the dog's immune systems? Did their bodies fight back against the virus?
Yes, and this is expected. All the dogs developed neutralizing antibodies.
Neutralizing antibodies.
Yes, they're antibodies that basically target the virus. And this happened within 2 weeks of treatment. So in some cases, this might have actually limited how effective the virus was.
So their own immune systems were kind of working against the treatment in a way. That's the crux of the matter, isn't it?
Yeah, to a certain extent, yes. It's something we commonly see with these kinds of therapies. Future studies, they'll probably need to find ways to kind of, modulate the immune response, maybe combine rLAS-uPA with other treatments, or even tweak the dosing a bit.
That makes sense. Now, besides just tumor size, it sounds like they also looked at other factors. What about quality of life? What about neurological function? Did those measures show anything?
They did. There was a definite positive trend towards improved quality of life scores.
Really?
Yes, especially in those dogs that showed either shrinkage or stabilization of the tumor. And importantly, most of the dogs, they didn't have any major declines in neurological function.
That's great news. That means the treatment wasn't causing additional neurological problems, right?
Precisely. And that's always a crucial thing to consider in these cases.
Let's dig a little deeper into that immune response. You said they measured levels of certain cytokines in the dog's blood. What did those findings show?
Right. They looked at specific cytokines like TNFi, IFNR, and TRAIL. These are signaling molecules. They play a key role in how the immune system responds to infections and cancer.
Okay.
What they found was that the levels of all 3 of these cytokines, they increased quite a bit after the treatment. This shows that the virus was definitely stimulating the immune system.
So it's like the virus sends out a signal, And then the immune system troops rally, right?
That's a good way to put it. But this immune stimulation, it can be a double-edged sword.
How so?
Well, while these cytokines can definitely help fight the tumor, they can also be part of those side effects we were talking about, things like fever and inflammation.
So figuring out that balance, how to maximize the good effects and minimize the bad ones, that's the burning question.
Exactly. That's where future research needs to focus. And speaking of the immune system, this study also looked at the complement system.
Okay, tell me more about that. The complement system. How does it play in all of this?
Well, the complement system is a complex network of proteins. It's a crucial part of our body's defense against, anything that wants to cause us harm. But sometimes it can be a bit overzealous.
Overzealous how?
It might attack not just the bad stuff, but also things that are trying to help, like therapeutic viruses, like our rLAS-uPA.
Oh, so it's working against the treatment.
You could say that, yes. It can actually neutralize the virus before it even reaches the tumor cells.
Wow, that's a big problem. Did they find any evidence of that happening in the dogs?
Well, they didn't measure complement activation directly in the study. However, the fact that the neutralizing antibodies developed so quickly, it does suggest that the complement system might have been involved.
Okay, so that's another puzzle piece they need to figure out. What are some strategies they're looking at to make these therapies more effective, especially when it comes to this complement system issue?
One really promising approach is to modify the virus itself, to make it, less susceptible to complement attack. Maybe incorporate certain proteins like CD46 or CD55.
I see. So, give it a disguise so the complement system doesn't recognize it.
Exactly. These proteins act as a kind of shield, preventing the complement system from attacking the virus.
That's clever. Any other strategies they're exploring?
Oh, definitely. Combining oncolytic viruses with other treatments is a big one. Things like chemo or immunotherapy.
So like a team effort, hitting the tumor from all angles.
Yes, exactly. For instance, some chemo drugs can actually help the oncolytic virus replicate better within the tumor cells, so they boost each other. And immunotherapy, that uses the power of the immune system itself. That can also be a great partner for oncolytic viruses. Well, oncolytic viruses can trigger the release of what we call tumor-associated antigens. These are like red flags, alerting the immune system to the cancer cells. So combining them with immunotherapy, we could potentially make the immune system even better at recognizing and destroying those tumor cells.
It's fascinating. What about using oncolytic viruses in humans for brain tumors? Is that on the horizon?
That's the ultimate goal, of course. And these findings from the Canine study, they give us valuable information that we can use to design human clinical trials.
Are there any trials going on right now using these viruses in humans for brain tumors?
Yes, actually there are several. One that's particularly noteworthy is using a modified herpes simplex virus to treat glioblastoma.
And how are the results looking so far?
It's still early days, but there have been some encouraging signs, both in terms of effectiveness and safety.
So there's definitely hope on the horizon.
Absolutely. Oncolytic viruses hold immense potential to, truly revolutionize cancer treatment. And this study in dogs, it's bringing us one step closer to making that potential a reality. And speaking of potential, this research also emphasized the importance of something called UPA expression.
We talked about the UPA receptor before, but why is the expression level so important?
Right, so UPA, it stands for Urokinase Plasminogen Activator-Receptor. It's basically a protein found on the surface of many cell types and, importantly, some cancer cells.
And why is this receptor so crucial when we're talking about these viruses?
Well, the rLAS-uPA virus, the one they used in this study, it was specifically engineered to target that UPA receptor. Think of it like a lock and key. The virus can only infect cells that have that specific receptor on their surface.
So it ensures that it's only targeting the tumor cells and leaving the healthy cells alone.
Exactly. And what they found is that tumors with higher UPA expression, they were more likely to respond to the rLAS-uPA treatment.
So could UPR expression be used to predict which patients are more likely to benefit from this type of therapy?
That's the hope. If we can identify patients with high UPR expression, then we can give this therapy to those who are most likely to respond to it.
Like personalized medicine, right?
Oh.
Tailoring treatment based on each patient's tumor?
Precisely. And that's where cancer treatment is heading, moving away from that one-size-fits-all approach and towards more targeted, personalized therapies.
Now let's talk about the challenges involved in doing these clinical trials with oncolytic viruses. What are some of the biggest hurdles?
Well, one of the main challenges is manufacturing these viruses. Producing them on a large scale, it's a very complex and expensive process.
I can imagine.
And then, of course, there are safety concerns. With any virus, there's always a risk that it could mutate and become harmful.
So rigorous testing is key.
Absolutely. And researchers are always working to make these viruses safer, but there are also regulatory hurdles. The rules and regulations for oncolytic viruses, they're still being developed.
So it's a bit of a waiting game while the science and the regulations catch up.
In a way, yes. But the progress is remarkable, and I'm confident that we'll see many more of these oncolytic virus therapies becoming available in the near future.
Now let's focus back on this specific study. We've talked about tumor shrinkage, but what about other ways to measure how well the treatment is working? Did they look at things like survival rates or quality of life?
Yes, they did. Now, this study wasn't designed to look at overall survival, but they did look at what we call progression-free survival. This is how long a patient lives without the disease getting worse.
Okay, and what did they find?
The results were actually very promising. The 6-month progression-free survival rate was 88%. This means that 88% of the dogs in the study were still alive after 6 months, and their tumors hadn't grown significantly.
That's impressive, especially when you consider that, brain tumors usually have a pretty poor prognosis.
It is. And remember, these dogs were only treated with the rLAS-uPA. They didn't have any other treatments, like surgery or radiation.
So the virus was doing all the work, essentially.
That's what the data suggests. It really shows the potential of these oncolytic viruses.
What about quality of life? Did they see any improvements in the dog's overall well-being?
Yes, they did see a trend towards improvement in the quality of life scores. And these scores, they were reported by the dog's caregivers, so people who knew them best.
That's really good to hear. So it sounds like the dogs were living longer and feeling better.
That seems to be the case, yes. Of course, more research is needed to confirm these findings and to find ways to make this treatment even more effective and safe.
You mentioned earlier that they also looked at the dog's neurological function. Why is that so important?
It's important because brain tumors can often cause problems with the nervous system. Things like seizures, weakness, or even changes in behavior. The good news is that in this study, most of the dogs didn't experience any significant worsening of their neurological function after the treatment.
So it seems like the treatment wasn't harming the brain any further.
Exactly, and that's a very important thing to consider when you're evaluating any treatment for brain tumors.
So overall, this study paints a pretty hopeful picture for oncolytic virus therapy for brain tumors.
I would agree. There are still challenges to overcome, but the potential benefits are huge. Now, let's step back and think about the broader implications of this research. What does it mean for veterinary medicine as a whole?
This research, it has the potential to change how we treat brain tumors in dogs. Right now, our options are often limited.
That's right. rLAS-uPA could be a new and potentially more effective option for treating our canine companions.
And because canine brain tumors are so similar to human brain tumors, these findings could have implications for human medicine as well.
Exactly. It's a win-win situation benefiting both dogs and humans.
Now let's talk a little bit more about this rLAS-uPA virus. You mentioned that it's a modified Newcastle disease virus. Can you explain that a bit more?
Sure. So Newcastle disease virus, or NDV, it's a type of virus that infects birds, particularly poultry. It's actually a very common poultry virus. But certain strains of NDV, well, they've shown promise as what we call oncolytic agents.
Meaning they can kill cancer cells.
Exactly. And what's really remarkable is that NDV, it seems to have a natural preference for tumor cells. It tends to infect and destroy cancer cells while leaving healthy cells relatively unharmed.
That's incredible. So why did they modify the virus for this study?
They modified it to make it even better at killing tumor cells and to make it safer for use in dogs. So they engineered it to target that uPA receptor we talked about earlier, which makes it more specific for tumor cells. And this can reduce the risk of it infecting healthy cells.
So it's like they're giving the virus a GPS, guiding it directly to the tumor cells.
That's a great analogy. And they also modified it to make it less likely to cause disease in dogs, so it's safer for them.
That's good to know. So they essentially took a naturally occurring virus and tweaked it to create a more effective and safe cancer-fighting agent.
Precisely. And this approach has shown promise in other studies as well. It really highlights the potential of using viruses to fight cancer.
It's truly remarkable what scientists can do these days.
It is. It's a real testament to their ingenuity.
Now let's talk about the immune response to this rLAS-uPA virus. You mentioned that the dogs developed neutralizing antibodies. Can you explain what that means and why it's important?
Sure. So neutralizing antibodies, they're like special proteins produced by the immune system, and their job is to, well, bind to and inactivate viruses.
So they're like little soldiers that target and destroy the virus.
That's a great way to think about it. And in this study, the dogs developed these neutralizing antibodies within 2 weeks of the treatment.
And why is that important?
It's important because it means that the dog's immune systems were able to recognize the virus and mount a defense against it.
Which is a good thing, right?
It's good and bad. It's encouraging that their immune systems were able to respond to the virus because it means the treatment is stimulating the immune system. But these neutralizing antibodies, they can also limit how effective the treatment is because they can inactivate the virus before it has a chance to kill all the tumor cells.
So it's a balancing act, right? Trying to harness the power of the immune system without letting it sabotage the treatment.
Exactly. And that's one of the big challenges in developing oncolytic virus therapies.
Now let's talk about the potential side effects of rLAS-uPA. We know that it was generally well tolerated in the dogs, but were there any side effects that we should be aware of?
Well, as with any treatment, there were some side effects observed in the study. The most common ones were pretty mild, and they didn't last very long. Things like fever, chills, and sometimes diarrhea.
Those sound like the kinds of things you might experience with a mild cold or flu.
That's a good comparison. And usually these side effects resolved on their own within a few days.
Were there any more serious side effects?
There were a few, but they were rare. Only 2 dogs experienced more serious side effects, which were thrombocytopenia and cerebral edema.
Can you explain what those are?
Sure. Thrombocytopenia means that the blood has a low platelet count. Platelets are what help your blood to clot, so if you don't have enough of them, you're more likely to bleed, and cerebral edema is swelling in the brain.
And were these serious side effects definitely caused by the treatment?
It's possible, but we need to do more research to be sure.
And did the dogs recover from these side effects?
Yes, they did. Both dogs recovered at the right medical care.
That's good to hear. So it sounds like the potential benefits of rLAS-uPA outweigh the risks, at least based on what we've seen in this study.
That's a reasonable conclusion, yes. But as with any new treatment, it's important to monitor patients carefully for any potential side effects.
Now, what's next for rLAS-uPA? What are the next steps in terms of research and development?
Well, one of the key things we need to do is figure out the best way to give the treatment. What's the best dose and how often should it be given?
And what about addressing those neutralizing antibodies we talked about?
Yes, that's another big focus for future research. Scientists are looking at different ways to overcome this problem. One idea is to modify the virus so that it's less likely to be attacked by the antibodies. Another idea is to combine rLAS-uPA with other treatments that can temporarily suppress the immune system, which would give the virus more time to work.
It sounds like there's a lot of exciting work ahead.
There definitely is. This field of oncolytic virus therapy is moving very quickly, and there's a lot of potential to make a real difference for both animals and humans. And the study, it's a big step in the right direction.
Now, before we wrap up, I want to touch on something that's really important in any kind of medical research, the ethical considerations, especially when we're talking about animal research.
Absolutely. Ethics should always be at the forefront of our minds.
What are some of the key ethical considerations in this type of research?
Well, first and foremost, we need to make sure that the animals are being treated humanely. Their well-being should always come first.
Were there any specific steps taken in this study to make sure the animals were treated ethically?
Yes, definitely. The study was conducted according to very strict ethical guidelines, and everything was approved by an institutional animal care and use committee.
So there was oversight to ensure the animals' welfare.
Yes, exactly. And the researchers also took steps to minimize any potential discomfort or distress that the dogs might experience.
That's reassuring. It's important to remember that these animals are making a real contribution to scientific progress, and we have a responsibility to treat them with respect and compassion.
I couldn't agree more.
Now let's bring this deep dive to a close with some final thoughts.
This study has opened up some really exciting possibilities for treating brain tumors in dogs and potentially in humans as well. It shows us what we can accomplish when we combine scientific ingenuity with compassion for animals.
And it reminds us that even when we're facing a tough disease like brain cancer, there's always hope for new and better treatments.
Absolutely. The future of oncolytic virus therapy is very bright, and I'm really excited to see what the future holds.
I'm sure our listeners are too. So from Vet Neurojar, keep those minds inspired, hearts light, and tails wagging.
This could really be a game changer for how we treat these brain tumors in dogs. Right now, the options we have are often limited, And they don't always work very well. This rLAS-uPA, it could be a real breakthrough for our furry friends.
It's definitely promising. And because these canine brain tumors, they're so similar to human brain tumors, could these findings have implications for human medicine as well?
Absolutely. It's a win-win situation, really, and, potentially helping both dogs and humans. That's what's so great about comparative oncology research. We learn from each other and hopefully develop treatments that benefit everyone.
Let's dig a little deeper into this rLAS-uPA virus. You mentioned that it's a modified Newcastle disease virus. Can you tell me a bit more about that?
Sure. So Newcastle disease virus, or NDV for short, is a virus that you typically find in birds, like poultry. It's actually quite common in poultry. However, certain strains of this NDV, they've shown some really promising results as oncolytic agents.
So meaning they can kill cancer cells?
Precisely. And what's fascinating is that NDV seems to have like a natural affinity for tumor cells. It prefers to infect and destroy cancer cells while leaving healthy cells relatively unharmed.
That's pretty incredible. So why did they modify the virus in this particular study?
Well, they modified it to, first of all, enhance its ability to kill those tumor cells, and secondly, to make it safer for use in dogs. So they engineered it to target that UPA receptor, which we talked about earlier. This makes it much more selective for tumor cells, and that potentially reduces the risk of it infecting healthy cells.
But a more targeted approach.
Exactly. And they also made some modifications to make it less likely to cause disease in dogs, so, making it even safer.
That's reassuring. So essentially, they took this naturally occurring virus and fine-tuned it to be a more effective and safer tool for fighting cancer.
That's a great way to put it. And this approach, it's not just promising in this study, it's showing promise in other areas of research as well, really highlighting the potential of harnessing the power of these viruses to fight cancer.
Let's shift gears a bit and talk about the immune response to rLAS-uPA. We've discussed these neutralizing antibodies before. Can you unpack what they are and their significance a little bit more?
Okay, so neutralizing antibodies, you can think of them like specialized warriors that your immune system produces. Their job is to target and deactivate viruses.
So they seek out the virus and eliminate it.
Exactly. And in this study, the dogs developed these neutralizing antibodies within just a couple of weeks of the treatment with rLAS-uPA.
Is that a good thing or a bad thing?
Well, it's a bit of both, to be honest. On one hand, it's encouraging because it shows that the dog's immune systems are recognizing the virus and trying to fight it off. It suggests that the treatment is stimulating the immune system, which could have some added benefits.
But on the other hand,
the antibodies, they could also limit how well the treatment works because they could potentially deactivate the virus before it has a chance to really do its job and kill all the tumor cells.
It's a tricky balance then. So how do researchers plan to address this?
Well, one promising strategy is to modify the virus even further to make it less susceptible to these neutralizing antibodies. Another approach is to combine the rLAS-uPA with other therapies that can temporarily suppress the immune response. This would give the virus more time to work its magic.
Okay, now let's turn our attention to the potential side effects of rLAS-uPA. We know it was generally well tolerated, but were there any notable side effects?
Yes, as with any treatment, there were some side effects, but the most common ones were pretty mild and they were temporary. Things like fever, chills, sometimes diarrhea. These typically went away on their own after a few days.
Were there any that were more serious?
There were 2 dogs that had more severe side effects, thrombocytopenia and cerebral edema, but those were rare cases.
And do we know if those were directly caused by the treatment?
It's possible, but we need to do more research to confirm a direct link.
And did they recover from these side effects?
Yes. Both dogs recovered with proper medical management.
That's good to hear. So it seems like, based on the study, that the potential benefits of rLAS-uPA, they outweigh the risks.
Yes, that's a reasonable conclusion. But of course, as with any new treatment, it's important to be cautious and monitor for any potential side effects.
What's next for rLAS-uPA in terms of research and development?
Well, a big focus now is on figuring out the best treatment protocol. What's the optimal dose? How often should it be given? Those sorts of things.
And what about the challenge of those neutralizing antibodies?
Right. That's another crucial area for future research. Scientists are exploring different ways to overcome this, modifying the virus to make it less susceptible, or maybe combining rLAS-uPA with other therapies that can help modulate the immune response.
It seems like a very exciting time for veterinary neuro-oncology. The potential of these oncolytic viruses is really remarkable.
I agree. This research represents a significant step forward in our fight against these brain tumors in dogs. And the hope for new, more effective treatments, well, it's incredibly encouraging.
Let's talk about the idea of personalized medicine using this virus.
That's one of the most exciting things about this research. It opens the door to personalized medicine. Remember how the study showed that tumors with higher UPA expression responded better to the treatment? Well, in the future, we could use a simple biopsy to assess a dog's UPAR expression levels before we even start the treatment.
So that way we can identify which dogs are most likely to benefit.
Exactly. It allows us to personalize the treatment for each individual dog, maximize the chances of success, and minimize unnecessary side effects for those who might not respond as well.
That's incredible. It's like taking precision medicine to a whole new level for our furry companions.
It really is. And this idea isn't limited to just UPR. As we learn more about these viruses and how they interact with different tumor types, we can identify even more biomarkers that can help us tailor treatment strategies.
So what are some of the big questions that still need to be answered?
The immune response is still a big one. Figuring out ways to either temporarily suppress the immune system or to engineer these viruses so they can evade those neutralizing antibodies, that's going to be critical. And then there's the question of dosing and timing. Is one big dose better, or are multiple smaller doses over time the way to go?
Those are really important questions. What about combining this therapy with other treatments?
Right. That's another avenue we need to explore. Combining rLAS-uPA with chemotherapy or immunotherapy could really enhance its tumor-killing abilities.
It's like putting together a dream team of cancer fighters.
Exactly. And we also need to think about the long-term effects of this therapy. It's not just about how well it works in the short term, but also about making sure it's safe in the long run.
So long-term monitoring and follow-up are essential.
Absolutely. Long-term studies are going to be critical to fully understand the potential benefits and risks of this treatment approach.
It feels like we're on the verge of a new era in veterinary neuro-oncology. The possibilities with these oncolytic viruses are pretty amazing.
I wholeheartedly agree. This research is a major leap forward, and it brings a lot of hope for new and better treatments that can improve the quality of life for our beloved pets.
And before we conclude, I just want to touch on something that's often overlooked in medical research, but is so important. And that's the ethical considerations, especially when we're talking about animal research.
Right. Ethics should always be our top priority. We have to make sure that any research involving animals is done ethically and humanely.
Absolutely. So were there any specific measures taken in this study to address ethical concerns?
Yes, definitely. The study followed very strict ethical guidelines, and all the procedures were approved by an institutional animal care and use committee. The researchers took every precaution to make sure the dogs were treated humanely and to minimize any discomfort or distress.
That's good to hear. It's important to remember that these animals are making a real contribution to science, and we owe it to them to treat them with respect and compassion.
I completely agree. Their well-being should always be our primary concern.
This has been a really insightful deep dive. What final thoughts would you like to leave our listeners with?
This research is incredibly promising. It shows what we can achieve when we combine scientific ingenuity with a deep compassion for animals. It gives us hope for new and more effective ways to treat these devastating brain tumors, not just in dogs, but potentially in humans as well.
This has been such a fascinating look into these oncolytic viruses.
It really has. In this research, it gives us so much valuable information about their potential for treating these canine brain tumors.
Yeah, it's given us a lot to think about, like the way these viruses target those tumor cells. And then, the challenges we face with the immune response.
And the potential for personalized medicine. It's really exciting. Imagine, tailoring treatment for each individual dog based on their specific tumor.
It would be revolutionary for how we approach these canine brain tumors.
Absolutely. And this research, it could have implications for human medicine as well.
It's amazing to think that what we learn from studying these viruses in dogs could lead to breakthroughs for treating brain tumors in people too.
That's the beauty of comparative oncology. It shows how connected we all are, really.
So where do we go from here? How do we take what we've learned and actually turn it into real treatments for dogs?
Well, we need to keep building on this research. More research is needed, to optimize the treatment protocol, figure out the best dosing strategies, and of course, find ways to overcome those defenses that the immune system throws up.
It's like we're on this quest to unlock the true potential of these incredible viruses.
I like that analogy. And it's a quest that's definitely worth pursuing. The potential benefits, both for dogs and potentially for humans, they're just too significant to ignore.
This deep dive has been truly eye-opening. Any final thoughts you want to leave our listeners with?
This research, it's a real beacon of hope in our fight against brain tumors. It shows what's possible when brilliant scientists combine their expertise with compassion for these animals. And it reminds us that even with a challenging disease like this, there's always hope for finding new and more effective treatments.
From Vet Neurojar, keep those minds inspired, hearts light, and tails wagging.